morning, everyone. Welcome to the Arthritis web seminar series. Today's our lecture number 24 on osteoarthritis and genetics. Today's guest speaker is Dr. John Loughlin from Newcastle University in UK. He's going to be talking on osteoarthritis and genetics. Before we start the introductions, just a reminder to the audience, please post your questions on the question answer tab on Zoom. Over to you, Rob.
Thank you, Mohit. Welcome everyone. It's a delight to welcome Professor John Loughlin to our seminar series this morning. John did his PhD in developmental biology at the University of Leeds and postdoctoral studies at the Institute of Molecular Medicine in Oxford. He then went on to actually establish a group, the Wellcome Trust Center for Human Genetics at Oxford, and played a key role there. In a leadership role in the genetic analysis of osteoarthritis. He was awarded tenure at Oxford in 2002, and then 2008 moved to Newcastle University, where he's currently professor of musculoskeletal research. John's research group focuses on identification, characterization of OA risk-conferring DNA polymorphisms using a wide variety of new techniques in silico analyses, correlating the epigenome with genetic risk, and CRISPR-Cas9 technology to modulate these risk-conferring alleles. John, it's a delight having you with us today. Welcome.
Thank you very much. Thank you, Robert, and thank you, Mohit. So, as Robert mentioned, this is a kind of academic journey for me. I was born, raised in Leicester, in the middle of England, and Leicester's most famous for burying the last English Plantagenet king below a car park, and that person was Richard III, discovered about 10 years ago. After A-level studies in Leicester, I moved to Liverpool and did a degree in applied biochemistry, and that involved a year in industry as well. So I worked for ICI, which is now AstraZeneca. I then moved to Leeds to do my PhD. And at Leeds, I worked on homeobox genes in Xenopus borealis, so a lot of kind of molecular biology work. And then after that, I then moved to Oxford to start my postdoctoral studies. And those were working with Professor Brian Sykes, and I was investigating initially single gene disorders of the musculoskeletal system. And in Oxford, I then moved on to investigating osteoarthritis. And then in 2008, I moved to Newcastle, where I'm currently based, and have a group, as Robert mentioned, that investigates the genetics, epigenetics, and functional analysis of osteoarthritis risk. So for those who don't know, Newcastle University is a red brick university situated in the northeast of England. And just some figures about the university: it was established in 1834 as part of Durham University, and it was doing the School of Medicine and Surgery for Durham. It has 25,000 students, 18,600 undergrads and 6,400 postgrads. There's approximately 2,400 academic staff. And we have three faculties: humanities, sciences, and medical sciences. And I'm a member of one of the medical sciences three institutes, the Biosciences Institute. And this institute is doing the kind of discovery level medical research, trying to understand the molecular basis of life. And within the Biosciences Institute, I'm then a member of what's known as the Skeletal Research Group. And this is a group that has five principal investigators: Cassie, Eloise, Mike, myself, and David. And we all investigate different molecular genetic aspects of musculoskeletal diseases, some single gene disorders, some polygenic traits such as osteoarthritis. So that's a bit of background. So I'm going to be talking to you today about osteoarthritis and genetics. And what I've done is I've broken my talk up into three components. So current status of genetic understanding of OA, functional analysis of risk alleles, and then I'll end talking about how we could actually use some of this OA genetics research. So I'll start with the current status of our genetic understanding of osteoarthritis. And when you think about doing a genetic analysis of a polygenic trait, a disease, something like osteoarthritis, you've got to think really seriously about, well, what is it that you're really investigating? What form of disease? And in a way, there's three types of osteoarthritis that, as a geneticist, one could come across. The first is early onset forms of osteoarthritis that are caused by rare mutations. So these are things that occur in skeletal dysplasias, such as pseudoachondroplasia and multiple epiphyseal dysplasia. So these forms of OA are early onset, caused by a mutation that has normally a catastrophic effect on protein function. The other form of osteoarthritis that one could come across are the non-genetic forms. So situations where somebody's had a severe trauma to their joint, so they therefore develop osteoarthritis. Often these could be sports-related effects, and these individuals could be relatively young. So as a geneticist, you would try and avoid these if you're trying to understand the genetic causes of osteoarthritis. And then the form that we do investigate is that common age-related form of osteoarthritis. So we know that it's familial, but it lacks an obvious cause. It's not caused by a trauma or anything like that. So this is the form of OA that I and many other people investigate when we're trying to understand the disease. And this form of osteoarthritis is characterized by the gradual loss of articular cartilage. So in this diagram, this is a hip removed from somebody who had severe osteoarthritis, and they've got this full thickness lesion. So this person would obviously have been in a lot of pain, and they will have gone forward for joint replacement surgery. And the osteoarthritis that we investigate principally affects the hands, knees, and the hips. Now, obviously, other tissues are also involved in the OA disease process. It's not just cartilage. And we need to bear that in mind whenever we do any investigations of this disease, thinking about effects on other tissues. And the nature of the genetic risk for this common form of osteoarthritis has been studied to a large degree using things such as twin studies and segregation studies, as well as other epidemiological investigations. And what these have told us is that this common late onset form of osteoarthritis is non-Mendelian, it's complex, and it's multifactorial. So to try and identify risk alleles for this form of osteoarthritis, we have to undertake genome-wide association studies, GWAS, and we do this by testing millions of single nucleotide polymorphisms. So the idea here is that there's a polymorphism that correlates with the disease. Its frequency is slightly elevated in individuals with the disease versus individuals without the disease. So it's not absent in the controls, but it's increased frequency in the cases. And there have been a lot of osteoarthritis GWAS studies undertaken over the past decade. The vast majority of these have been performed in Europeans or individuals of European descent, so often North Americans. Meta-analyses have then been undertaken. And the idea there is that you're trying to combine as much data as possible so that you have maximum power to see those subtle differences in allele frequencies between your cases and your controls, particular polymorphisms. And the largest GWASs performed and published to date have over 70,000 cases and 370,000 controls. So those numbers are quite reasonable for picking up these subtle differences in allele frequency. And the current state that we're at is that there's about 124 OA risk loci have so far been reported in the literature. When you take these together, they account for about 25% of OA heritability. There are no common risk alleles of large individual impact. So we don't find effects as we do in, for example, rheumatoid arthritis or ankylosing spondylitis at the HLA, where you can have a particular locus that has a large individual impact. Instead, most of the odds ratios for osteoarthritis signals are quite small, so OR is less than 1.5. And something else that has become apparent from the genetic studies is that there appears to be joint site heterogeneity. And what that means is that a risk allele for osteoarthritis at the hip won't necessarily also be a risk allele for osteoarthritis at the knee. So there is this difference between different skeletal sites when it comes to the genetic component of the disease. So OA is therefore highly polygenic. So if you take an individual with osteoarthritis and you've excluded, for example, trauma and severe trauma as the cause, and you have somebody with the late onset primary form of the disease, from a genetics perspective, their disease has come about because they've inherited a large number of risk alleles for the disease. So it's a very polygenic condition. So to summarize that, DNA polymorphisms, not mutations. Mutations are the things that we see in skeletal dysplasias, rare effects that are highly penetrant. In osteoarthritis, it's polymorphisms instead. You discover these through GWAS. The risk alleles themselves have relatively low odds ratios. There is this joint site heterogeneity, and it's a highly polygenic disease. So what I'm going to do now is move on to the functional analysis of risk-conferring alleles. So what's become very apparent, not only in osteoarthritis, but in all common complex disease studies, is that the GWASs have identified a very large number of signals of single nucleotide polymorphisms that correlate with the disease. But what we haven't done as much of is then taking those signals and trying to work out what is the causal polymorphism, what is the target gene, what's the actual functional effect, so what is happening here. One thing that has become apparent is that when you identify a polymorphism through a GWAS, so for example, if this is your GWAS SNP, it's not necessarily the causal one. There can be other polymorphisms that correlate with your GWAS SNP, and you need to try and identify that causal polymorphism itself. And the other thing is that these polymorphisms tend to reside within intergenic regions or non-coding parts of the gene. So most polymorphisms for most complex traits don't alter protein coding sequence. Instead, they regulate the expression of the gene, how much of the mRNA is synthesized, where it's synthesized, and so on. So it's a regulatory effect. Something else that's becoming apparent, and particularly so in osteoarthritis, is that there is an interaction with epigenetics, and in particular DNA methylation. So it seems to be the case that if you identify a GWAS SNP and a causal SNP, there may be an intermediate effect, and that particular polymorphism may alter DNA methylation in the intergenic region, and that itself will then regulate the expression of a gene. So if you take your polymorphism of interest and you measure DNA methylation in the vicinity of your causal SNP, you can generate what's known as a methylation quantitative trait locus, or an MQTL, in which the polymorphic alleles correlate with the levels of DNA methylation. And the assumption, therefore, is that the causal SNP alters local DNA methylation, and that this itself leads to alteration in expression of genes. So when doing these types of functional studies, what we're trying to do is to move from the GWAS signals through to understanding how the variants affect function. And then hopefully with that additional knowledge, we can then think about translational investigations. And what we do in our group is we do a variety of different analyses. So we take in silico data, things that have been published and are available publicly, and we try and work out from a GWAS signal what the likely target gene is, what the potential causal polymorphism is. And then we move into doing investigations within the lab itself. So we actually do directed experimental studies in the lab to see if we can actually elucidate what's happening between a GWAS signal through to function, and then perhaps potentially applying some translational insights. So to do this, I'm going to talk to you about a study that's ongoing in our lab at the moment. And this is work that's being done by a PhD student called Julia, and she's co-supervised by Dr. Sarah Rice. And the investigations that we do when we're moving from a GWAS signal through to trying to understand the functional effects is we use tissues from OA patients following arthroplasty. So in Newcastle, we're really fortunate in that we have very close collaborative links with the Freeman Hospital and the Royal Victoria Infirmary in Newcastle. And through those two hospitals, we're able to work with orthopedic surgeons and access tissue from patients who are having their joints replaced. So we can get things such as cartilage, but also synovium, subchondral bone, fat pad, and so on. So the study I'm going to talk to you about is an investigation into this particular polymorphism here. It's the SNP is RS11581583641. It's a C to T polymorphism, and this C is the risk allele for osteoarthritis. This is the p-value that was created from a meta-analysis of this study. And the polymorphism itself resides within the 3' untranslated region of COL-GAL-T2. And this gene encodes a collagen galactosyl transferase enzyme. So the gene itself resides on chromosome 1, 1q25.3. This is a diagram of the gene itself. And this is zoomed in, just showing the polymorphism within the 3' untranslated region. And what we discovered is that this polymorphism correlates with methylation changes in a predicted enhancer region of the gene. So this part of the COLGAL-T2 gene, which is intronic, was predicted as an enhancer based on in silico data. And we identified a number of CPG sites within the region that show methylation correlations with the polymorphism. This one highlighted in green is the CPG that we discovered this signal from the 450 Illumina methylation array. And what we did is we correlated methylation in CPGs flanking this CPG9 to assess how wide this methylation quantitative trait locus was. And this is the data here. And what each of these shows is data for patients that are homozygote CC for the risk allele, heterozygote, and then homozygote TT for the non-risk allele. And as you move through it, you can begin to see that there is a correlation between genotype and methylation of CPGs, particularly this CPG 8, 9, and 10. So these CPGs are showing differences in their methylation status based upon correlation with the risk allele. And the risk allele leads to reduced methylation of these CPGs. So we hypothesized that the risk of this locus is potentially differentially methylating this enhancer, and that may have effects upon the expression of COLGAL-T2. So Yulia and Sarah then went and investigated this further. So one of the first things that we did is we took that enhancer region, we then cloned it into a vector, a CPG-free vector, transfected a chondrocyte cell line, the TC28A2 cell line, and we said, does that enhancer act as an enhancer in this chondrocyte cell line? And does it act as an enhancer when we methylate or demethylate it? And what we discovered is that yes, it does act as an enhancer. And if we methylate it, we reduce the enhancer effect. So this piece of DNA is methylation sensitive. When it's unmethylated, it has a stronger enhancer effect than it does when it's methylated. The next thing that we did is we used CRISPR-Cas9 technology to delete the enhancer. So the enhancer is located within an intron of COLGAL-T2, but there are flanking genes here as well. And we wanted to know that if we were to delete this piece of DNA, would it impact upon COLGAL-T2, or would it impact upon other genes that are flanking it? So we deleted the piece of DNA in the TC28A2 cells using CRISPR-Cas9. And what we discovered is that the deletion of it led to a reduction in the expression of the gene. So it is an enhancer for COLGAL-T2, but doesn't have an effect on either of the two flanking genes. We then used CRISPR-Cas9, but this time we used deactivated or dead Cas9, and we methylated and demethylated the enhancer. So we're not deleting the piece of DNA, but instead we're sticking the dCas9 to the DNA, and then we're methylating and then demethylating to assess whether differential methylation of this enhancer also had an impact upon COLGAL-T2. So this is a diagram just talking you through it. So we used a range of different guides to guide this piece of the dCas9 to the piece of DNA, the enhancer, within these cells. And in black, we see where we have the control, so we don't have the guides to take the dCas9 to the piece of DNA. And in the pink just here, we have where we have managed to take the dCas9 to the enhancer region, and we're able to, in this instance, methylate it. So we're going from a low level of methylation to a high level of methylation, and we managed to do that with a range of different guides very consistently. And on this side, we're demethylating. So we're using TET to reduce the levels of methylation. And what we discovered is that when we methylated this piece of DNA, it did have an effect upon the expression of COLGAL-T2. We were able to reduce the expression of the gene. And when we demethylated it, we were able to increase the expression of the gene. And the effect, again, was only seen on COLGAL-T2. We then went to our patient samples, and we identified individuals that were heterozygote for the polymorphism in the 3' UTR of the gene. And we said, if you're heterozygote, does the risk allele produce more or less mRNA than the non-risk allele? And this is an experiment known as allelic expression imbalance. And it's a means of identifying and expression quantitative trait lockers. So does a polymorphism correlate with changes in expression of the gene? And what we have here is each of these are individual patients that we've analyzed. In black is their allelic ratio in their DNA, so their 1-to-1 ratio. And in yellow here is the allelic ratio for the C to T polymorphism in their RNA. And these are technical repeats for each of the analyses. And what we noticed was that the C allele, the risk allele, led to increased expression of the gene. So we noticed that there was this allelic expression imbalance operating in patient samples. And these are cartilage samples from individuals that have undergone arthroplasty. So if we take that data together, what our current model is that this polymorphism in the 3' UTR of COLGAL-T2 is an association signal. It's been replicated, it's robust. It correlates with decreased methylation of the enhancer, which resides within an intron of COLGAL-T2. We believe that this leads to increased expression of COLGAL-T2, and that increased expression of this enzyme will lead to increased post-translational modification of collagen, and that this will presumably be detrimental to cartilage tissue homeostasis. Now, we're only partway through this particular study. What we haven't done yet is we haven't yet identified the actual causal SNP. We don't know whether this 3' UTR polymorphism itself is causal or is not. We haven't identified the transcription factors, which are presumably differentially binding to the enhancer region in response to methylation changes. And we also haven't yet identified the collagen target of the enzyme. We predict it's probably type 2 collagen and the main collagen in cartilage, but we don't have any evidence for that yet. So there's still lots of work to be undertaken on this, but using these molecular genetic studies enable you to move to transition from a GWAS signal through to function and to get a clearer understanding of what could be going on in a particular locus. The other thing that we're starting to do, and others have done plenty of this, is to take the signals that are identified through these genetic studies and to see where they reside in pathways. Do we begin to notice particular pathways are emerging from these types of studies? So for example, COLGAL-T2 is involved in collagen synthesis. So that's one pathway that does appear to be impacted in OA genetic studies. So you can look at different genes and different signals and see other examples where those genes and their protein products appear to be involved in collagen and proteoglycan synthesis. Other things that are becoming apparent are effects on transcriptional regulation, osteoblast development, chondrocyte development, cell proliferation, apoptosis, and also skeletal development and morphogenesis. So there's a lot of things that we're beginning to uncover on the basis of genetic studies that we're able to say that certain pathways appear to be particularly important in the disease. So to summarize that, OA genetic risk is regulatory. What we don't tend to see very often are polymorphisms that correlate with amino acid substitutions. Instead, most osteoarthritis polymorphisms, those that are associated with the disease, correlate with regulatory elements. So they're in intergenic regions, in promoters, in untranslated regions of genes, in introns. They're not in protein coding sequences. There appears to be this interplay with epigenetic factors, and this works in a number of different ways. So one example is the one I've shown you here, where a polymorphism appears to influence the methylation status of an enhancer. But other things that we're beginning to see is that some polymorphisms are involved in regulating microRNAs, other polymorphisms can be involved in regulating histones and so on. So there is that strong interplay between epigenetics and genetics in osteoarthritis. When we do these functional studies, that enables us to elucidate mechanisms. So using COLGAL-T2 as an example, we're able to start to get a handle upon how these polymorphisms are working and what pathways are emerging. And the pathways that are emerging appear to be quite diverse. So we're seeing a number of different effects occurring across a range of different pathways. So what about the utility of OA genetics research? What can we actually do with it? So what I've done here is I've just listed three potential possibilities. One is therapeutic insights, one is polygenic risk scores, and the other is tissue engineering using CRISPR-Cas9. So this is a table from a paper by Eli Zaghini and colleagues published a couple of years ago. And this is from one of the largest GWAS studies performed so far in osteoarthritis using the UK Biobank. And what Ellie and her team then did is, having got signals, genes that they think are potentially causal polymorphisms as well, to then say, well, are there any therapeutics out there that actually have efficacy that one could potentially apply to osteoarthritis? So some simple examples are TGF-beta1, where you've got Invosa, is a cell therapy that's possible and can be used and is under trials. GDF5, where you have protein that you can actually utilize, FGF18 and sprifermin. So there's plenty of examples now where we're getting signals from osteoarthritis genome scans, where you can then look to see what therapeutics are available. And that kind of gives us hope that potentially we could move towards applying some of these therapeutic insights in treatment of the disease. The other thing is polygenic risk scores, or trying to identify individuals that are likely to develop the disease, and maybe starting to get a handle upon when they'll develop the disease. So maybe some kind of ability to give people risk estimates. So the idea here is obviously that polymorphisms are very prevalent in the population. We're all walking around with polymorphisms for different polygenic traits for different diseases. And some of us will inherit a certain proportion of polymorphisms that mean we will develop that particular disease. So if you cross this threshold of liability, then you're likely to develop the disease. So using polygenic risk scores, we can start to say to individuals, you've inherited a certain number of risk alleles. Your likelihood of developing that disease is elevated in comparison to other individuals. Another means by which this could be of use is in clinical trials, where we're trying to identify cohorts in whom the disease will progress. So one of the issues in osteoarthritis is that sometimes you can develop a drug, try it out on a population, but the individuals take a long time to progress. So you're unable to quickly test efficacy. If we could identify people who are likely to progress, then we may have a better opportunity to intervene and therefore test efficacy of that compound. So that's the utility of polygenic risk scores. And then the other thing is tissue engineering. So if we're able to identify risk alleles for osteoarthritis, and then we're able to identify what those risk alleles are actually doing, are they altering expression of particular genes, can we then somehow intervene to try and overcome that effect, to attenuate it to some degree? And this, I suppose, is where the epigenetic side of things may be of particular value. If we know that a polymorphism is, for example, leading to increased methylation or demethylation of a region, then we may be able to epigenetically modulate a particular region and enhancer to alter the expression of a gene. And there's a variety of different tools that we could use. So the ones that I discussed here, such as TET and DNMT, but there are other enzymes that one can utilize as well with the deactivated Cas9. And then maybe at some point we can get to the position where we can overcome those deleterious effects of those risk alleles and perhaps make a better cartilage. But there is a note of caution here, and it's something that I did touch on earlier about pathways involved in osteoarthritis. One of the things that's becoming apparent and has been known about and reported on for many years now is the developmental origins of osteoarthritis. So I'm listing here some review articles that have been published from a range of different investigators that have flagged up the link between osteoarthritis, genetics and developmental biology. And also the work from Terry Cappellini and his team, where they're able to show, particularly the GDF5 locus, that there does appear to be an interaction between development and also osteoarthritis. So what that opens us up to is the need to seriously think about when these genetic effects are operating. So when we're identifying differentially methylated regions, effects on gene expression, when does that actually come about? And what we've been doing in the group is working with the Human Developmental Biology Resource, which is a Wellcome-funded, an MRC-funded study based in Newcastle and also in London. And we've been accessing tissue specimens from fetal samples. So we've been able to access cartilage from these fetal samples. And then we've been able to isolate cells, isolate nucleic acids. And we've then gone and had a look at some of the effects that we discovered in arthroplasty samples, so in old cartilage, and said, are these genetic effects, are these functional effects evident in these fetal samples? And I'll give you an example here. This is a methylation QTL analysis that we've been doing on one of the osteoarthritis genes that we have an interest in called PLEC. And PLEC codes for the protein plectin, which is a cytoskeletal molecule. And what we've discovered is that if we look at MQTLs in fetal samples, they're as readily observable as they are in OA cartilage samples. So we're picking up the same effects in developmental cartilage as we are in osteoarthritis cartilage. And what we do notice is sometimes there is an absolute difference in the overall methylation levels. So at this particular CpG here, the average methylation of the GGs is about 70%. And in the Gs in the OA cartilage, it's about 50%. So there does appear to be a demethylation in OA cartilage, but that genetic effect endures throughout life. So presumably it's there and established during development and then continues throughout life, but there is a slow demethylation at that particular locus. And that's just shown in this diagram here. What we've also done is we've taken other MQTLs that we've identified at OA risk loci, and we've done an analysis to work out effect size. So how much does genotype contribute to the MQTL effects that we observe? And for this, we've used developmental limb, developmental cartilage, and OA cartilage, and a range of different CpGs and a range of different genes. And what we do notice is that the effects often are stronger in developmental cartilage. So for some loci like this PLEC locus here, they're actually stronger in OA cartilage. But overall, we see stronger effects in fetal cartilage, in developmental cartilage, than we do in osteoarthritis cartilage. So again, that's kind of implying to us that a lot of these effects are laid down during development, that they endure through life, but that's something that we obviously need to think seriously about. So to summarize that, there's obviously broad scope for potential intervention based upon the genetics discoveries that are being made, but when to intervene needs a lot of thought. We obviously think about osteoarthritis as a disease of older people, but really it is a disease but has deep developmental roots. So when we're thinking about intervening in the disease, at least from a genetics perspective or a genomics perspective, then we do need to put serious thought to what would be the appropriate time point to do that. So just to conclude remarks on this, there has been huge progress in the mapping of risk loci for osteoarthritis. Genome-wide association studies have identified over 120 risk loci so far, and many more will be following in the near future. Functional investigations are gathering pace, so this is combining in silico analyses, but also direct laboratory-based studies. And these types of investigations require the use of cell lines, patient samples, and so on. There is scope for translation. It does appear realistic, but it's likely to be quite challenging, and that we're going to have to think about when we do this. But it is a very exciting time to be researching this area. And it's something that anybody who isn't particularly involved in the genetics of osteoarthritis, I think if you get involved in it now, you'll have an awful lot of fun and excitement. So finally, I just want to thank the funders who support my research, in particular Versus Arthritis, who have funded me for many years, and the MRC through a grant known as SEMA. The data generators in the group. So I've already mentioned Sarah and Yulia, who do a lot of the work on the Colville TT study, but the other investigators as well, Abby, Tony, Guillaume, and Jack. Also, David Amaza, who actually developed the CRISPR-Cas9 tools that we use in our lab. And that was at a point when these were quite novel, quite difficult to get working, and he did a fantastic job enabling us to do a lot of experiments that we now do. And also the Newcastle upon Tynes Hospital. So as I mentioned, the RVI and the Freeman and the orthopedic surgeons are fantastic in giving us access to the samples from patients. And finally, if anybody wants to know a bit more about these topics, we've recently published a review in Osteoarthritis and Cartilage on the genetics of osteoarthritis. And last year, we also published a review on that interplay between genetics and epigenetics in the disease. And please feel free to check those out. And if you have any questions relating to those, don't hesitate to contact me. Thank you.
Thank you, John. Terrific overview and great studies and great progress. John, we're just waiting for the questions to pop up on the chat line. Just a reminder to the audience, please post your questions on the question and answer tab on Zoom. John, I wanted to start off with the first question around tackling the early stage of the disease and really trying to link genetics, which predispose individuals to progress faster compared to individuals who would be progressing faster in the disease trajectory. Are there efforts which are being put, not only looking at the late stages of osteoarthritis, but focusing on early stages?
Yeah, I mean, there are quite a few studies underway, and we're involved in a study called Approach, where we have a cohort that we're tracking over 2 years, and we're doing a number of measures of those, kind of gait analysis, conventional biomarker analysis, but also genomic analyses. We're then using machine learning to see if we can actually identify individuals that are more likely to progress than other individuals. We've just finished that study. The month 24 data is now coming in, so we should have a handle on that at some point soon. But others are doing the same thing. There is that desire to try and see if we can identify rapid progressors so that we could do that. And I think genomics will obviously play a major part in that, because it makes perfect sense that if you're genetically loaded, you're more likely to progress and maybe progress in a shorter period of time. So yeah.
That's great.
There we go. We have a question from Amanda Alley. John, thank you for your presentation. What is the state of the OA field in exploring gene by environment interactions, specifically the effect of environmental factors impacting genetic risk in OA?
Yeah, I mean, there's lots of investigations using things like the UK Biobank and stuff like that, where people are starting to investigate those things. It's not something that I'm particularly involved in, but I do know that other investigators are trying to untangle what are those environmental components to the disease. And things do start to emerge. I mean, I know diets involved, smoking, alcohol, and stuff like that. So there are things emerging from those. So we are beginning to get a handle on that. But it's, in comparison to the genomic side of it, I think it's still at its early stages. But those large cohorts like the UK Biobank are really important for those types of investigations.
Next question is from an anonymous attendee. Often osteoarthritis arises from undiagnosed prior inflammatory arthritis. How do you try to exclude these from primary osteoarthritis?
Yeah, well, that's an interesting question, because the inflammatory component to osteoarthritis is quite heavily debated. It's a low-level inflammation, if at all, I think in a lot of instances. I know you can get erosive hand osteoarthritis, which probably has a stronger inflammatory component to it. What we're tending to find from a lot of the GWAS studies is that we're not necessarily seeing an awful lot of inflammatory mediators coming forward. We're seeing alternative things. So I'm beginning to wonder whether there's a perception out there that there is a strong inflammatory component, but in fact there isn't really, or it's misinterpreted or something like that. So, and to be honest, when we've done these studies, we try and avoid anybody that's got rheumatoid, ankylosing spondylitis, or any of the classic stuff. But apart from that, we don't dig that much deeper in the exclusion criteria to say you cannot come in if you've got a relatively modest inflammatory aspect to your disease and stuff. So we do try and separate out the inflammatory arthritis, but that's as far as we go, really.
John, I had a question at the sort of the strategy at population-based studies. We may have known risk factors. Obesity would be an example. How do you discriminate if there's an associate in a GWAS? You have a hit that's actually an indirect marker. You might have you might have a SNP, for example, that predisposes to obesity, for which OA is a consequence. And I wondered how you can tease out what are the intermediary rather than the primary relationships.
Well, the thing that we should remember is that a lot of risk alleles will be shared between different complex diseases. So sometimes you will be identifying risk alleles that are also risk alleles for other traits as well. So, you know, when it comes to teasing them apart, there are epidemiological ways of doing that. So you can factor in body mass index and stuff like that in your studies to try and exclude that. But don't think that just because it's a risk factor for osteoarthritis, it can't be a risk factor for some other trait. It can be. Lots of these are shared between different diseases as well.
Right.
John, you touched base on your comparisons with the developmental samples as well as from the OA samples. This is something which is emerging in the field, that there are endogenous mechanisms which could help in making our joints. But then these endogenous mechanisms sort of become pathological in nature, which are basically predisposing individuals to osteoarthritis. And you also mentioned this a study which was published in Cell by Capellini Group, and they've actually looked into with GDF5. Would you be able to expand on the link between development and osteoarthritis, and how does this process, which is homeostatic in nature, helps in development, but actually predisposes individuals to osteoarthritis?
So I suppose there's two things there. One is the recapitulation of, say, the growth plate or something like that. So you get hypertrophic cartilage and all of those kind of things, which is often what people focus on. When we're doing our fetal analysis, we're not really saying that's what we're seeing. What we're saying is that these genetic effects are laid down during development. So, and what kind of Terry is kind of saying is a similar thing. He says that the derived joint shape is evolutionary driven, and then you need to leave it like that. But you may have some polymorphism that subtly change that to some degree. That's tolerated during development because the derived joint shape is more or less still intact. But as you age, it becomes detrimental to joint health, and therefore it has a knock-on effect. And I'm more at that side of it. I think that we should think about what's happening during development in laying down your tissues and your shape and all of that kind of stuff that is then detrimental as you age, rather than thinking about a recapitulation of the developmental phenotype as you age. I think they're kind of separate things in a way. So and that's how we're viewing it.
So you would envision both the mechanical aspect as well as probably the genetics, and maybe there are other molecular mechanisms which would probably be at interplay in conjugate.
Yeah, absolutely, 100%. Yeah.
Absolutely. That's good.
Madi Hagigi asks, John, what do you think about the correlation between the MAPT gene and OA?
So with that question, what do you actually mean? What do I think about it? What does that actually mean? Do I believe it? Do I trust it? Am I excited by it? All of those, I believe it and I'm excited by it, but I'm not really sure what else to say.
Yeah, okay.
That's great. We're waiting for more questions to come, John. I read the review article on genetics and epigenetics, which was published in Nature Reviews Rheumatology from your group. And one thing which really intrigues us is how we can really utilize the expertise which are in genetics and also epigenetics. Are you taking the same samples which you're subjecting to the genetics analyses as well as GWAS, and then looking at the epigenetic regulation as well to really parse out the mechanisms here?
Yeah, I mean, we do what we do. So, I mean, a big core part of what we do is DNA methylation. So we correlate risk alleles with local DNA methylation changes in cysts, and then try and understand those. So in the way that I described with Cole-Gall. We're also interested in polymorphisms at or near alternative epigenetic regulators. So WWP2 is the host gene for MER140, which is a really important microRNA cartilage development, and it has a polymorphism. So we're currently trying to investigate it to see whether that polymorphism is actually altering this expression of MER140 or WWP2 or both. And then the same with histone regulators. There's things like NCOA3 is a gene that harbors OA polymorphisms. There's an EQ-cell at that locus that correlates with the signal, and NCOA3 regulates histones and stuff. So there's lots going on there from an epigenetics perspective. So it's either mapping to epigenetic regulators or correlating with or influencing to some degree. But I think the neat thing about epigenetics, I suppose, is that you can kind of intervene. You can think of models where you could go in organoids and all of this kind of stuff and start to have a play. It just seems a bit more straightforward than trying necessarily to alter the actual coding, the genetic sequence, the actual DNA-based changes and stuff like that. So that's kind of why we're kind of interested in it more so.
Our experience, John, looking at spondylitis GWAS has been plagued in many studies by gene admixture. I mean, Newcastle may be like Toronto in this sense, but Toronto is a remarkably multiracial, multi-ethnic genetic mix. Sure. And now sometimes that can be a real headache for the genetic epidemiologist, but I wondered also if there's some opportunities there. Are there pockets, racial ethnic pockets that seem to be relatively resistant or more susceptible to OA?
Yeah, I mean, there are differences. There appears to be differences in ethnic risk allele frequencies and stuff like that. So, you know, if you compare some of the Asian GWAS studies that have been performed to European GWAS studies, you do see different signals emerging, as well as lots of shared signals. So, as Mohit touched on before, GDF5 is the original GWAS, original OA risk allele signal that was discovered in Asians and has been replicated in Europeans. But at the same time, Asian studies have identified a number of other signals that we've never really replicated in Europeans. So there are ethnic differences between groups. That's definitely the case. So the architecture will be different between different groups.
Right.
John, are you using other joint tissues as well? You mentioned fat pad or synovium.
Yeah. We do. We analyze as many tissues as we can get from patients and stuff, and we look within a patient as well. So when we see NQTL effects and EQTL effects, we try and compare and contrast within tissues. And others are starting to do this as well, or have been doing this as well. So we kind of aren't cartilage centric as such. We try and broaden our view to a large degree. But what we tend to find is that if we see a signal, it is at least in cartilage and probably is in other tissues as well.
Right. And do you look at systemically as well? So looking at circulation?
Well, we do have blood samples from some of our patients. So we often, I mean, it's so relatively far removed that if we do see an effect in cartilage, we tend not to see it necessarily in blood. And there's loads of in silico data for blood as well. So you can get a lot of EQTL data for blood, methylation data.
In terms of validating, I guess there are lots of challenges with in vivo validation, but I wondered, John, what the status of looking at mirroring models, say of something like the COL-GAL-T2 modifications in a mirroring model, how that's...
Yeah, no, there are plenty of people who do animal models for osteoarthritis, the DMM model and all of this, and I've started to look at particular genes and stuff like that. And again, Terry's work on GDF5 enhancers downstream of the gene and stuff like that has been exceptional. But at the same time, if you feel you've got the variant or you believe you've got the function, then I don't think there's anything better than using human samples. Right. I mean, just get on with it, you know. So I don't, you know, but if you haven't, or if you want to do more subtle stuff, I get why people do use the animal models. But for the work we do, we're more than happy to use the human samples to do that type of work. I mean, we're the best model system going, I think, for this.
Right. We don't want to cure murine OA in the final analysis.
Well, if you read the literature, it's cured about once a week in the publications. Somebody's always publishing that they've, you know, stopped arthritis in the DMM model with a intervention or something like that. But yeah.
Right.
That's great. John, no more questions on the chat. Thank you so much for joining us today. It was a great lecture and great discussion.
That was wonderful. Thank you very much, John. That was wonderful.
You're very welcome. You're very welcome.
And thank you to the audience for joining us today. Please join us on April 27th on lecture on targeting the CD40 pathway in Sjogren syndrome by Dr. Benjamin Fisher from Birmingham University in the UK. See you all on April 27th. Again, thank you so much, John, for joining us.
Bye, John.
You're welcome. Bye-bye. Bye-bye.